everyone, welcome back to my channel Agrotech once again. Please guys keep following me on Facebook, blog account and Twitter, and please keep loving my videos. Today I'm here to give you a lecture about polymerase chain reaction, which is called PCR in short form. And if you guys haven't like and share my channel, please do like and share my channel, and please do not forget to subscribe my channel before going to the content. Then let's move on to the actual content. What is PCR then? Guys, the DNA, which is highly tiny and very small. And for this study purpose, we cannot extract heavy volume of DNA at all time. We only can make or extract DNA into the very small volume. So to carry out a lot of experiment regarding that DNA from the small volume, we can use PCR. You know, PCR is a polymerase chain reaction which allow to copy or amplify thousands or many DNA copies. So from a small volume of DNA, it will allow to make it enough for a study. That's why PCR has an important role in genetic engineering or biotechnology study. It is the only tool like make us easy to study the DNA, characterizing DNA or anything related with DNA and RNA. It makes us easy to study from small volume of DNA and RNA. Then, how it works? Yes, it has a specific way to work. Initially, the DNA we have used to amplify used to get denatured, which means the DNA strand used to break into the single strand. Double-stranded DNA break into the single-strand DNA, and we have to use the forward and reverse primer before setting the PCR reaction. So these forward and reverse primer got annealed into the single strand DNA after certain stage. Then after certain stage, the primer used to extend towards the single strand and finally make double strand complementary DNA. And these steps, initial denaturation, annealing, extension, and final extension, happens within the PCR reaction, and we have to hold that reaction at the last. And how we got result is we got result in this way. We use the initial DNA fragment to be amplified, and at the stage of denaturation they got break into the single strand DNA. Then our forward and reverse primer got annealed into the single strand DNA. Then the elongation happened there at this stage. The primers got stick towards the or in anneal towards the each strand of DNA got extended, and then after certain cycle we will get certain copies of our original DNA. It called complementary DNA. We got complementary DNA from our original DNA, and after certain cycle we would get enough volume of DNA that would be enough for further study. And there is a specific procedure to amplify DNA and get multiple copies. Mean there is a specific procedure for PCR. The circular DNA itself cannot be amplified because it is too lengthy to make complementary DNA. So what we have to do first is we have to identify a certain fragment of DNA or certain length of DNA to be amplified. From the whole length of DNA, only the certain length of DNA, the length of DNA can be measured in base pairs, so certain base pair length of DNA can be amplified. So first we have to identify that much length of DNA to be amplified from the whole circular DNA or a whole genomic DNA. Then we have to identify the forward and reverse primer for that section of the DNA. Then we have to add nucleotide bases because the DNA sequence is a nucleotide base sequence. Therefore, we have to add nucleotides so that they can anneal with the single-stranded DNA after denaturation. So we also have to add polymerase. With the help of polymerase, these nucleotides would get stick to the complementary DNA strand and make complementary double-stranded DNA. So all the reaction should be on a specific mix buffer. So in a mix buffer in a PCR tube, we have to add a certain fragment, sorry, the whole DNA, sample of whole DNA. Then we have to identify the primers. We have to add the primers there. These primers will amplify the certain section of the DNA. Then with the addition of nucleotides and the DNA, sorry, and the polymerase, the polymerase help to bind the nucleotides on the strand, and the forward and reverse primers will help to go with a long chain and make a complementary strand. So in this way we make PCR reaction. Then what we have to do, we have to put these reaction in a PCR machine. We can make the PCR reaction in small tubes, like I have shown here in the black image with the small white tubes. These are the specific small size of PCR tube if we have the small volume or small size of reaction. And if we have larger volume of the reaction, what we have to do is we can make the reaction on 90 well plate. We call this 90 well plate because it has this small 90 wells. We can make the 90 different reaction there. So we can place these tubes on the PCR machine, and we have to set up the program. Usually we follow the same denaturation temperature for all of the reaction, but the extension or elongation time would be different based on the the segment of DNA to be amplified. How long is our segment, more would be our extension time, more would be our elongation time. So based on that, we set up this system, PCR program after reaction for at least more than 20 cycles. Then after 20 or 20 to 40 cycles, we would get enough volume of DNA. This is complementary DNA segments from the original DNA. And you can see the small tubes with the green liquid. This is the PCR reaction made in a small tube. The green is because of the polymerase. We nowadays we generally use Taq polymerase because it has the GFP plus all of the DNTPs. So it's a most common polymerase using in the PCR reaction these days. Then after after we set up the PCR program, once we have the the whole PCR reaction after running the setup program, what we have to do is we have to visualize the result. Otherwise, we cannot confirm the segment is amplified. Then how do we visualize is, after we got the PCR tube from the PCR machine, we take them out and we run these DNA in agar gel, or sometimes we can run in acrylamide gel as well. So after running the DNA sample with the loading marker, after running for maybe 1 hour or half an hour, we can see the bands on gel after putting them onto the UV chamber. Then we would see this kind of result, the bands and the marker, and with the help of the marker we can identify our right band size there. Then we can confirm if the right size band appears there, then we can confirm we have the very good PCR result. In this way we verify the result. And guys, this much for today. And if you want to know more about the other aspects of biotechnology, other aspects of agricultural biotechnology, please stay tuned with my channel. So we will see you on next video. Till then, stay healthy, stay hygiene, and keep loving my videos, guys. Thank you.